A very warm good morning to everyone and welcome to the first international guest lecture in the academic year 21. And we have Ankita Raikar with us, who is our alumnus, and it's so proud to introduce you in this session. And as Ankita sent me her picture, I picked her picture up from the LinkedIn profile. So currently, she is working as a scientist, clinical biomarker innovation and development, Takeda Pharmaceuticals, USA. And let me tell you, she has an impressive resume for her 8 years of stint in the pharma industry. She was always been a bright student, maybe top 5% of her class. And we always knew that she would go into research. And it is so nice to see her translate into a hardworking and efficient scientist. She's grown up the professional ladder, and here I share her professional experience with you. So she's joined Novartis Institute of Biomedical Research as Scientific Associate 2, moved up the ladder to Scientist 1, post which she is at her current position in Takeda, and she also worked as a scientist, cellular pharmacology, in EMD Serono. So that's a nice series of professional experience that she has gathered over the years after her primary education of she did her master's in pharmaceutical sciences, Northeastern University, Boston, Madison, USA, and worked on HIF1 alpha activation in 3D tumor spheroids. So just a quick introduction to what is 3D tumor spheroids is essentially these are three-dimensional mimics in cellular assays. So typically we work in two dimension when we do these in vitro anti-cancer screening assays. So 3D tumor assays are typically more connected to the live cells that are there in the system, and hopefully they mimic more closely the responses versus the 2D model. So a lot of people shifting on to 3D models for their in vitro studies. And she, of course, has a bachelor's in pharmacy from a college. So over to you, Ankita, and a very, very warm welcome from all of us over here. We are so happy to have you on board with us today.
Yeah, thank you so much, Tabassum ma'am. Like this, thank you so much for the kind words and a very nice introduction. And also thank you for giving me this opportunity. This is kind of my first as well, like lecturing the BPharm students. So it's like a really nice opportunity for me as well. So thank you for considering me for this particular lecture series. And I have made a few slides, so I'll share it with you guys. And it is, and I want to keep it more informal. So please, feel free to stop me at any point to ask questions or, even, and I don't want to make it too scientific. This is just to kind of give you an overview of like what I've been doing and kind of an introduction to the world of biomarkers if you're not already aware of it. It's just like a high level overview, and then we can talk about if you have any specific questions about career in the US or, pursuing your master's, PhD, or in a specific field, I can probably, based on my experience, try to help you or guide you through that. So I don't know if you can see my screen. Uh,
Yes, we can.
and is it like a full screen, or do you see it like in a,
No, it's
I
full
don't,
screen. It's perfect.
it's okay,
Go
it's
ahead.
fine. So, all right. So this is, I had a, slide for just going over my career trajectory, but I think Tabassum ma'am, you did a great job at that. So I won't spend too much time here, but I just wanted to kind of give an overview of like what I've been doing in each of these roles that I've put years. So, like, of course, I did my bachelor's at BNCP and then moved on to pursue my master's at Northeastern in pharmaceutical sciences. So, I did my master's in a drug delivery field. It was very like nanoparticles, liposomes focused. And what I'm doing now is completely different. It's still research, but I would say I've evolved through like these years as a scientist from like one field to other, and it's been like a really nice like experience for me as well to go from something from drug delivery into like preclinical research into clinical. So while I was doing my master's, I got this opportunity to do an internship at a mid-sized biotech company in Boston called Infinity Pharmaceuticals, and here I kind of got exposed to the world of cancer biology. I was in the biology department. I got my first exposure into the, like working in the lab and, working on all these different experiments where we are focusing on PI3 kinase inhibitors and its effect on, in vitro cells and trying to, figure out the potency and stuff like that. And then I was trying to look for a full-time job and ended up at Novartis as a scientific associate too. So this group was heavily focused on bispecific antibodies in oncology. So here I was involved in the field of oncology, preclinical research. And when I say preclinical research, this was right from the conception of a target, like, a viable target that is good for a specific type of cancer, to all the way to putting these studies into non-human primates like monkeys, where you test these drugs in monkeys. So I mean, I wasn't involved in it, but just like pushing the drug from its conception all the way to the non-human primate studies. And here I spent almost 5 years at Novartis. And since this was all preclinical, I didn't get much exposure on the translational or the clinical side of things. And I really felt that, I know how to work on a bench now, and I really wanted to take my expertise and learn something more on the clinical side. So I started looking for jobs and found this really nice position at Takeda, which was completely on the clinical side. So this is like heavily, I would say, clinical focused biomarker centric job. And here I directly get to, play a role in the clinical trial. So it's quite exciting. It's only been a year so far, so I wouldn't say I'm an expert, but I'm still learning. So, but I love it on the clinical side of things. I'll probably continue on this side. So, so for today's presentation, I just wanted to give you a brief introduction, like what are biomarkers, like, how are they classified, what are the different technologies or platforms that we use for biomarker measurements, how are these assays developed whenever we want to use this assay in a clinical study, they are going to be used for making a decision for that particular drug, like in the clinic. So how are we developing these assays to make it fit for purpose? And I'll also touch upon like a case study or an example that I'm working on and how that biomarker data is actually used to make decisions in the clinic. And lastly, I would just go over some of the future perspectives in biomarker research. And feel free to stop me anytime if you have any questions. Okay. Yeah, so I've made this like cliched introductory slide on like what are biomarkers. So I am not familiar like how much you have been exposed to this area, but I just wanted to start quite basic, like, so biomarker is like a abbreviation for a biological marker. So it is anything that can be measured in a human body that can tell you about any normal biological process, or it could tell you about a disease or the progression of the disease, or it could be used for something, like as a response to how the drug is acting. Like, is it is it helping to improve like the patient's health or not, so it could be something as simple as like measuring blood pressure for hypertension, or like even like measuring your own body temperature that tells you that you have fever or not. So something as simple as that is a biomarker. But in the clinical trials, like biomarkers are heavily used now to make a lot of decisions, and these decisions could be about the dose or the dosing schedule or whether the drug is acting or not. So biomarkers play a very, very important role in clinical trials. And majorly, they are kind of classified into like there are different categories. So I would say that it could be of physiologic origin, so it could be like measuring your own body temperature. It could be radiographic, like x-rays, ct scans. It could be histologic, like whenever you go to a pathologist and they take your blood smear on a slide, it is like a histologic measurement. Molecular, like measuring enzyme levels or a certain gene mutation, those are all kind of biomarker measurements. And like in today's world, I would say that they play an important role not only in the clinic but right from the preclinical stage, and it helps guide a particular drug right from its initial preclinical time to all the way to the last phases of the clinical trials. I would say even after the drug is approved. Um, I just want to make sure that you can hear me. I can't hear any feedback. So,
We can hear you.
okay, great. All right. So then I picked up this figure from the papers, that what is like an ideal biomarker, because often, like there could be tens and thousands of things you can measure whenever a drug goes into the clinic, or if it's going into the clinic. But you have to be very careful, like what you're measuring and what is the biomarker doing to help you in any particular decision. And oftentimes, like we see with these clinical trials in oncology, they are so expensive. Like you will see like all these biomarker measurements from one single patient cost as high as like $100,000. So you have to be very careful about like which biomarkers you pick. Are they helping you make any kind of decision? So that's why, like any biomarker you choose has to be cost effective. Like is it going to help you make some decision, or inform you anything about the drug? Any assay you're developing to measure the biomarker has to be, I wouldn't say easy, but it should be convenient. It should be reproducible. It can be performed in different labs. So you have to look into those kind of things as well. Like what are the platforms that you can use for measuring the biomarker? And so basically, you need to justify whenever you pick a certain biomarker, like what is it informing? Like, how is it going to influence your decision in this whole, clinical trials, or making a decision about the drug? Um, so next, I just wanted to give a brief introduction on like what are these different kinds of biomarkers, like how can they be classified? And this is like a nice figure summarizing like what are the different, what categories of biomarkers can be classified into. And I'll just go into somewhat like details of each of these categories with some examples, just so that it's easier to understand. So one of the categories is diagnostic biomarkers. So it is something that can be measured just to inform whether that person has a disease or not. So it could be something like blood sugar level to say that a person has type 2 diabetes, or in recent times, I would say like the COVID test, right? So when you measure the virus titers through a PCR test, you know that you have COVID or not. So something as simple as that, and it is like a widely used diagnostic biomarker. It's a widely used biomarker because we use it in our day-to-day lives as well, right? If you are sick, you would want to go to a doctor, they prescribe a few tests. So you have probably gone through like using a diagnostic biomarker yourself. So The second one is a monitoring biomarker. So that could be something that informs you about like how the disease is progressing. Um, and one good example is like looking at, say, blood concentration levels of addictive drugs in drug addiction prevention and treatment. Like, is the drug level going down in a patient as as he is going through a particular rehabilitation, for example? Or if you think about efficacy of an antiretroviral therapy, like you can, in HIV, you can measure the HIV RNA. So this can be classified as a monitoring biomarker. The next category is PD, or pharmacodynamic or response biomarker. So this is like heavily used in most of the clinical trials. If you want to know the mechanism, if the drug is following a particular mechanism of action, or like whatever is the mechanism of action of the drug, can you measure it? Like how effective is it? And this particular category is widely used to make decisions on doses or the dosing schedules. So this is like, I would say, in most of the clinical trials, you'll always see like a PD or a response biomarker, which informs more on the mechanism of action of the drug. The next category is predictive biomarker. So nowadays you might have heard a lot about like personalized medicine, or a certain, especially in oncology, if you are familiar with that disease area. Now with the advent of immunotherapy, you would see that some patients respond well to a drug, some patients don't. So these predictive biomarkers help in identifying patients who might be good, like good responders, whereas others might be non-responders. So, for example, there are certain genetic mutations in certain tumors which make them better responders to a treatment. So you can always screen for these biomarkers, especially in oncology trials, to inform whether a particular patient population responds or not. Um, and just the last three categories, prognostic biomarker, I would say it's quite similar and very confusing to predictive biomarker. I had to read a little bit to distinguish between those two, but it seems like a prognostic biomarker is something that you don't really need a drug intervention, but you can just predict like what is the outcome of a particular disease, whether it's progressing, whether it has stopped, like, something, a biomarker that informs you about like the disease condition or the progress, that can be classified as a prognostic biomarker. And again, like some certain genetic mutations can tell you if a particular patient will relapse, or if they have a higher chance of certain cancer, so those kind of biomarkers help in these decisions. The next category is a safety biomarker, and this is most widely used in phase 1 clinical trials. And because whenever a drug goes into the clinic and it goes in phase 1, the first criteria is to look at the safety of the drug before even you go to efficacy or potency or anything else. So something that informs you if the drug is toxic or not can be classified as a safety biomarker. And in recent times, like I work a lot in the field of oncology, so for something like immunotherapy, which has like a very high chance of activating your immune system, and that can cause an issue. So there is a specific cytokine called IL-6, which can be used as a safety biomarker to monitor this cytokine release syndrome in patients who are on immune therapies. So very important biomarker, I would say. And then again, the last category is a risk biomarker, which basically tells you whether you have a higher susceptibility to a certain disease. Again, the BRCA1/2 mutation, if you probably have that mutation, you're at a higher chance of developing breast cancer. So these can be classified as like in the category of risk biomarker. So I know this is a lot of information, but I thought like maybe giving the right kind of examples might help you understand better. So moving on, so now that you know that, okay, there are these whole, like series of biomarkers that you can measure, The next step is to identify what are the um, technologies or platforms that you can use for these biomarker measurements. So usually when a program or a particular drug is going into the clinic, there's like a team who sits down and they develop a biomarker strategy that these are the biomarkers we want to measure, and they can be informing you about different things. They can tell you about the activity of the drug. They can tell you about the safety of the drug, like if the patient is responding to a treatment or not. So all these, so you usually have like different kinds of biomarkers for any particular program or a trial. So the next step is like to identify how are we going to measure these biomarkers, like what kind of platform can we use. So so for example, if you are, and they can be classified into these 6 different categories. So one of them is like looking at genomics, transcriptomics. I would just pool them in one category. So they are looking at whether the particular patient has a genetic mutation or not. So you can measure it by, like an experiment using PCR, which is polymerase chain reaction. And nowadays there are like these next generation sequencing, like high throughput platforms that can give you basically like it can inform you about all the genes present in your genome. So, and how the drug is affecting this gene expression levels. Is it going up? Is it going down? Like, what is the effect of that expression levels um, on the immune system or the overall performance of the drug? So basically this is, there are like various different platforms you can use to get information on the genomics part of the study. The next part is like proteomics or cytometrics. So this is basically measuring any kind of protein in your say body fluids. It could be plasma, it could be serum, it could be blood. So there are different ways of measuring, and probably you guys are familiar with something as simple as ELISA. So there could be plate-based protein assays. And again, here the technology has advanced a lot. Nowadays, we have multiplex assays where you can measure 40 proteins in a single well. So it's really advanced right now. So that really helps. So you don't need a lot of sample from the patient. You can just take 100 microliters of plasma or serum and test for 40 different proteins. So these are like all high throughput screening assays that are quite used nowadays. And the other category is cytometrics. So I would say flow cytometry or mass cytometry, where you can look at different immune subsets. For example, if you are working on a particular oncology immune therapy, you might want to see how the drug is activating your particular immune subset. Are the T cells getting activated, or are the myeloid cell populations getting activated? So all of this can be easily studied using flow cytometry. And one of the newer technologies is mass cytometry, which is a combination of flow cytometry and mass spectrometry. So again, these are very high throughput. You can do as big, you can do lot of, from a single sample, you can get a lot of data. So that is pretty valuable, heavily used these days in clinical trials. And then you have imaging, like as I said, if you go to a pathologist, they take a blood smear on the slide, and then you have tumor biopsy. So that all comes under imaging. So that can also inform you whether a particular protein is expressed on the tumor or not. So that all comes under imaging. One of the newer, I think, uh, platform I would say is metabolomics. I'm not quite familiar with that. I would say it's something to do with studying the metabolites in the body and how do they respond to the drug. So my expertise in this is like I focus on proteomics and cytometrics. So I'm quite heavily involved in developing and validating assays to measure these biomarkers using proteomics and cytometrics platform. We also have several people in our group who are genomics experts, imaging experts. So basically, it's a combination of different expertise in these different platforms. Um, And then I just wanted to quickly go over like now that okay, now you have your list of biomarkers. You know what technologies you want to use to measure those. The next step as you go into the clinic is how are you going to develop an experiment or an assay that is consistent, that is reproducible. Every time you measure a clinical sample from the patient, it will perform the same way. So that requires a little bit of validation in the sense. And this is where preclinical and clinical research differs. And this graph here, I think it shows that pretty well, because so as you go from discovery and more into the clinic, the stringency of how you develop and validate these assays becomes quite, the stringency increases. Say in a preclinical setup, you would probably design an assay, test it quickly in the lab, and you get the results, and that is totally okay. But when you have to develop something for the clinic, you have to be very cautious that the assay you're developing is fit for purpose. It will perform the same way every time a patient sample is tested, and that requires a little bit of thoroughness. So you have to basically look whenever you're developing a particular experiment or an assay, you have to make sure that it has, it can detect that particular biomarker with the same accuracy, with the same precision. It is reproducible. It has, it has a particular dynamic range. What is the expected dynamic range? How sensitive is the assay? So all these things need to be tested, and they need to be documented somewhere. And this is extremely important because all these reports go to FDA whenever the drug is, whenever they file for the approval. So there can be an FDA audit, and they'll say, we want to see, how did you validate this data? Or how is the assay performing? Is the data reliable or not? So we have to do our due diligence and go through all these things whenever we develop this any assay for the clinic. And again, this flow chart here, it basically takes you right from preclinical to post licensure, like after the drug is approved. And how do you, like, what is the level of stringency required for each assay as you move from preclinical to phase 3, and then post licensure. So yeah. And then just to summarize, like whatever I have talked to you about now, like I just wanted to say that how the flow happens for this whole biomarker research or development. So basically, as I mentioned, like the first step is to, whenever a program or a drug decides to go into the clinical trial or start a clinical trial, the first step would be, what are the biomarkers we want to measure? So once you have the list of biomarkers, then you'll decide on what platform or technology you'll use. How will you develop the assay? What lab will you use? Because at Takeda we outsource everything, so nothing is done in house. We have CROs or contract research organizations who do this work for us. So what we have to do as subject matter experts on each of these platforms is to make sure that the CROs are developing the assay correctly, they are doing their due diligence, going over the reports that they generate. So that is how we operate, at least at Takeda. I cannot say for other companies. Each company has its own structure, but for us, it's mainly outsourcing these assays to other contract research organizations. And then once and whenever the platform, the lab, and everything is decided, you would work with them to develop an assay that is completely fit for purpose. It is basically adhering to all the validation parameters. You'll generate a validation report, and then finally, this assay will be used in the clinic to like test the patient samples, and then this patient data goes in the FDA filings, and to make important decisions for that particular molecule. um, so I gave you a lot of theoretical, I guess, information on biomarkers, but I also wanted to give like one example, and this is a program that I'm working on, it's an already approved drug. So this is this is all public information. And like I'm working with this drug now that it's approved. It's also, it's also used for some other trials or some other indications. So that is a different like area altogether. But what I wanted to show is Takhzyro is a drug that is approved for hereditary angioedema. And what happens in patients with hereditary angioedema is like they have this I think it's a rare genetic disease. So what happens is they have this mutation in their gene, which causes like a super high accumulation of this plasma kallikrein, which is an enzyme. Usually it is regulated by this, I don't know if you can see my, okay. Usually it is regulated by this C1INH protein, but in these patients, there is like a deficiency or low levels of C1INH. So what happens is like this plasma kallikrein is super activated, and uh, that causes like cleavage of this protein called cleaved high molecular weight kininogen, and it cleaves that protein and releases bradykinin. And what happens when, so bradykinin is a very potent vasodilator, and that is what causes a lot of edema and pain in these patients. So these patients often get these these inflammatory attacks, and it's very I would say it's unexpected. They can get it anytime, anywhere, and it can range from mild to very severe symptoms. So you will see like super high inflammation in the face or the limbs, anywhere. And then in extreme cases, it can also cause swelling of the larynx and cause death because of asphyxiation. So it has like a wide range of patients, ranging right from mild symptoms to like severe symptoms. So this drug has been, it's like a breakthrough drug. It has been given like an orphan designation, and it was it's like widely used by these patients. So what I wanted to highlight here is like when this drug like goes to the clinic, right? And you want to see if the drug is working or not. So one of the most important endpoint, or what would you look out for, is how is it affecting these HAE attacks? So if the patient is taking the drug, is it reducing the attacks? So that is like a very important endpoint, that okay, we have to, that is like the clinical endpoint, I would say. And then to support that, can we look into some of the PD biomarkers that, okay, now this particular dose of this drug is reducing the number of attacks. How can we combine this with the mechanism of action data or the PD data and say that, okay, this is the, everything, there's a good PK-PD relationship, and, this is the best dose. So I wanted to show some data that came out of this study. And this is like a phase 1 data, and they have looked here. So they had 3 different doses and at different schedules. So 150 mgs every 4 weeks, 300 mgs every 4 weeks, 300 mgs every 2 weeks. And they have looked at PK/PD relation, in the sense they are looking at the plasma concentration of these drugs in probably the patients enrolled in this trial. And then they are looking at this cleaved high molecular weight protein. So going back, if you look at the mechanism of action, right? The the drug actually inhibits this pcal. So technically it should it should inhibit the conversion of this protein it should prevent the breakdown of the protein, basically. So they are measuring this cleaved protein as one of the biomarkers to see what is the PD response to this drug? Is it doing what it's expected to do? So if you compare all these 3 doses, you see that the highest plasma concentration is reached with 300 mg given every 2 weeks, and that correlates very well to this dotted line. That is the dose that is giving the maximum suppression of this cleaved protein, indicating that out of those 3 doses, this one is it's probably the most effective. And then they have used this data also to look at the number of attacks in these patients. So So the lines that you see are the plasma concentration of the drug, and the bars you can see are the number of attacks. And again, these are three different doses, like 100, 150 mgs, 300 mgs, 300 mgs given every 2 weeks. So if you look at this particular graph, right, you see that with 300 mgs given every 2 weeks, you see the maximum plasma concentration, and also the number of attacks is going down better than the other two doses. So this is how they basically use the biomarker data along with the clinical data to make a decision on the dose or the dosing schedule, which one is the best. And then based on this data, they made a decision that for the phase 3 trial, they'll be using the 300 mgs given every 2 weeks. So this is just like a real life example that I wanted to give, like how biomarkers are used in most important decisions for the drugs. And and I would kind of like to end this I mean, at least this presentation, by talking a little bit about what lies ahead, like what is the future of biomarkers. So this is a really interesting concept of digital biomarkers, and I'm sure everyone is familiar with apps these days, like health apps or smartphones that collect all this data. And a lot of I would say a lot of companies are also investing some time and efforts in creating user friendly apps that can help make patient life more easy, or how they can track the data better. So I was just looking at and some of the companies that are working are Roche or Eli Lilly. They have departments who are more focused on digital biomarkers. So I was just going through like Roche's website yesterday, and I came across this like app they are developing where the patient can log in, like if the patient is suffering from certain disease that causes pain, so they can each day log in their pain rating. So based on a scale of 1 to 10, so that they have a record of how did they feel throughout the month. So when they visit the doctor, they have this record, because oftentimes when you visit a doctor, say once every month, you don't remember what days you had like severe pain, or when was it mild. So this was like an easier way to kind of keep a track of that record. So a lot of companies are now working towards developing these kind of apps, to help the patients measure their, and how can they basically use this data for informing about that particular drug. But of course, there are certain challenges where it's still primitive. I wouldn't even say that digital biomarkers are probably used in the clinic yet, because you have to figure out how are you going to standardize these measuring of digital biomarkers, because with different apps or different devices that you use, there'll be some variation, so there won't be certain amount of reliability. So that is a concern. You need to figure out how are you going to validate these particular platforms to perform exactly same each time they are used. And obviously there's a concern about privacy, data protection, because you might share some of your personal data with that company, but you don't know if that data is being shared with a third party company. So those are some challenges that I think it's still a far way to go. And the next thing I wanted to highlight was personalized medicine. So as I mentioned before, like a lot of immunotherapies are moving towards personalized medicine, because one drug fits all doesn't hold true these days, especially in oncology, because based on a particular genetic makeup of that tumor, a particular patient may not respond to a certain treatment, but may respond well to something else. So I felt that, this, like, a lot of companies are heading towards putting more research and more efforts in figuring out like what is the best way to move towards personalized medicine. And I think biomarkers play a really important role here. So yeah, so that is, I think, like two of the criteria that are gaining a lot of importance in the biomarker field. So yeah, so with that being said, I would like to kind of end my formal presentation. And if you have any questions, I'll be happy to answer those. I think you're on mute. I can't hear you. Sorry.
Yes, I am. Okay. So, thank you very much, Ankita, for taking us through the biomarkers journey. And I like the way you said most of you must be familiar with digital biomarkers. And I was like, oh, I don't know. What are these? Okay. So I think it was an interesting conversation. It also gave a new research perspective because you substantiated it with a case study and it showed a very good connect with the dose and the biomarker measures, which is actually the deliverable from most of the clinical endpoint studies. Right. So thank you very much, Ankita. And over to you, audience, if you have any questions, any question under the sun.
Yeah, it doesn't have to be biomarker specific. It could
Yes,
be
so it
pretty
could be
random as
anything.
well.
It could be for
Yeah.
M-Farm project. It could be life in US. It could be education in US, work in US. So it could be anything. So anybody wants to ask, please go ahead.
Hello, ma'am.
Yeah, go ahead.
Ma'am, my name is Bhushan Kumar. I'm pursuing masters from DY Patil IPS of Pimpri, Pune. Ma'am, my question is, what is the role of biomarkers in HPTLC biotography technique?
Sorry, I didn't catch the last part.
In HPTLC biotography technique, the role of biomarkers means
Uh,
how can we use?
Can I
so
can I answer that question?
Yes, ma'am.
yeah,
Yes, ma'am.
sure, because I am not
Yeah,
actually
okay. So
familiar
I'll
with
just
it.
tell you,
Yeah.
most of the times the techniques or the analytical methods which Ankita was mentioning, you have to validate the analytical methods. So 99.9% of the times you go in for HPLC or LCMS kind of studies to measure all this. HPTLC is generally not used because it doesn't eliminate the background sample interference which walks in, which HPLC can do very efficiently after suitable sample preparation techniques. So generally we do not use HPTLC because it doesn't give you a mass of the compound. So when we are doing metabolite profiling or clinical trials, we do need the mass of compounds, which comes very easily from LCMS. Hope that answers your question.
Okay, thank you, ma'am. Thank you. And ma'am, just another question is that how can we select the particular biomarker for the particular drug for particular activity?
Yeah, I can take that. So that depends on like it depends on what drug it is, what indication it is, whether you know the mechanism of action of the drug. For example, the one that I showed, right? It's a PCAL inhibitor. So we knew the pathway of how the drug acts. So can you measure something downstream of the drug where the drug acts that can be a good biomarker? And it doesn't, every time it may not work. You have to, for example, in the example that I showed, like bradykinin was like an ideal biomarker, because that is the reason why you see all this edema symptoms. But the problem with bradykinin was it's highly labile. It's really difficult to measure it. So we had to think about something else. So so then it was like something more, similar on the same pathway, but other than bradykinin, which was the cleaved high molecular weight kininogen. So even that gives a good idea of like how the drug is acting. So I would say it totally depends on what drug you're looking at, what is, if you know the pathway or not, like what indication it is. So it will vary based on each drug or molecule. That's what I would say.
Ankita, as an extension to this question, I would ask if you have a basket of, let's say, 5 or 6 biomarkers, how do you select?
Yeah, so that is a good question. I would say a lot of factors go into the selection process, and it is not that you have to stick to one. You can measure multiple. Many a times we do multiple biomarkers because the more the better. But oftentimes you have to look into how easy is it to validate it. Like you can start with validating all, but maybe in the end you'll only have one that is like a good one. You also have to look at the cost, because it often happens in most of our oncology trials that we want to measure everything. We want to measure cytokines. We want to measure look at tumor biopsies, and it significantly increases our cost. Like each patient is almost $100,000 cost. And I've been learning this a lot from like the global head in our department. So she insists a lot on think about why you are measuring it. What is it going to inform you about, and then go towards the selection, rather than just saying, oh, I want to measure all these 5 biomarkers. And it's always good, but you have to also look at the cost perspective as well. So yeah, so I would say I'm still learning how to do it, but this is some advice that she gave that I think is very valuable, that like always ask yourself why, what, and how. Like, why are you doing this? So yeah.
Do we have any more questions from the students? They have not changed much from your batch. They still don't question.
It's a
Some things
quiet
are
one.
very consistent, right? And the precision is very, very high. This is one of them. So students, if you want your photographs on the college Facebook and LinkedIn profile, please put your videos on, because I'm going to click a picture. Yeah, okay. So we got a lot of photograph takers, huh? Good. Okay. Vasanti, one second. Okay, so just hold on. Oh, one second. Okay, so I got it. And students, if you have any questions, this is a very good platform for you to ask and get your doubts clarified.
Or like, you can even reach out to me on LinkedIn later on. That's totally fine, with any questions you may
Yeah.
have. Yeah.
Yes, so we can have that. I can also share the Ankita's email address with you in case if you want to interact with her later down the road, right? So thank you very much, Ankita, and you adhered strictly to the time, because the students go in for a 10 o'clock lecture, and it's 10. So it was great having you here, and I look forward to being connected with you as our alumni time and again. Let's have some kind of interaction, because it keeps the connectivity on. Although we have connected after a decade,
Yeah.
let this be a beginning of a stronger bonding, with you participating in some of our college events as technical sessions, guest lecture sessions. Maybe you could be part of some course where you could, cover a little bit of clinical trial part. We have a course on pharmacovigilance. So maybe we could factor you in as one of our, resource persons for that. So thank
Yeah,
you
sure.
once again, Ankita, and it was a pleasure listening to you and sitting on the other side of the table. Okay, so normally we are on the other side of the table and you all are sitting there. So it was nice and there was a learning for me as well. Thank you so
Yeah.
much and have a wonderful weekend, Ankita.
Yeah, thank you so much for giving this opportunity to me. I enjoyed it a lot too. And I hope to stay in touch with you now that we have initiated, the talk. So yeah, please, like, feel free to
Great,
email me whenever you want. Yeah. And yeah.
great. So Ankita, I'm going to show you something.
Is that our batch? Like,
Yes, that's your
oh,
badge.
Is that your favorite batch? You have to admit that today.
Yes. ठीक
Maybe
है.
when I come to India, like I'll come visit you. It's been a
Yes,
while.
definitely. That's
Yeah.
that's mandatory now.
Yeah. Of
Okay,
course.
so I'm going to end the meeting now, and all of you can go back to your classes, and Ankita can go back and have a dinner, maybe, right?
Yeah. All
Okay,
right.
great.
Thank you,
Thank you
ma'am.
so much,
See
Ankita,
you.
and stay in touch.
Yeah. See you. Bye bye.
Bye.